Here. My name is David Regan, and I'm Chief Executive Officer of Sona Nanotech. I'm pleased to have with me here my colleague, Dr. Carmen Giacomo-Antonio, who is our Chief Medical Officer. Sona Nanotech is a nanotechnology company involved in life sciences, and we are aiming to do nothing short of transforming and lifting the current standard of care for those suffering from solid cancers. And we're doing that by empowering immunotherapies by stimulating the immune system. This is our tagline here, our novel 1-2 punch against cancer. We're going to explain that a little bit further, and we're going to dig into some tremendous preclinical efficacy results we have for our colorectal data that were released in a press release this morning. Our preclinical data suggests that when combined with standard immunotherapies, which is the standard of care used in cancer patients in most cases these days, examples of which would be Keytruda and Opdivo, Sona's targeted hyperthermia therapy can eliminate tumors. Now, will these promising preclinical results translate into human? These are, of course, the third tranche of our preclinical studies, the third type, and we'll cover off quickly some of the results we've done to date. We believe they will translate, and we're racing to get into the clinic this coming year to determine just that. But with a third set of preclinical studies, efficacy showing strong results, we're extremely encouraged by it. You're going to hear a little bit today about the different types of tumors, and what we talk about is cold tumors and hot tumors, and what exactly it is we're trying to accomplish. But with any further ado, let's dig into it. So we'll be making some forward-looking statements today, so we'd refer you to our disclosure documents and the cautions therein, of course. We're going to cover 4 areas today. We'll briefly go over what is targeted hyperthermia therapy, and I'd encourage those who are new to the story. There we have previous webinars on YouTube that you can go back and do a deep dive into. Secondly, we'll go through our preclinical results to date, just to bring everyone up to speed on those if you haven't had a chance. And then thirdly, of course, we'll dig into these results from the colorectal cancer model that we have. And finally, close off by giving a little bit more sense as to where we're going and what that timing might look like. Here's the problem we're trying to solve. Now, results from immunotherapies like Keytruda and Opdivo can be extremely powerful. They, the Nobel Prize was awarded for immunotherapies when it was introduced in about 2011. It's something that has transformed many lives. But the reality is, response rates are still low to those treatments. And I personally was shocked to see that. So in this slide, you see we've got the colorectal elements, where effectively you've got about 1 in 3 patients who respond to immunotherapy. So while it's incredibly powerful, you just want to hope you're one of those 1 in 3 folks. So what does Sona bring to the table in this challenge? We've got our patented biocompatible gold nanorods. What's a gold nanorod? Tiny piece of inert gold. And to give you a reference for the size, a single human hair has like 75,000 nanometers thick, just to give you a sense as to what that's like. They're very small. It's near atomic scale. Why rods as opposed to nanostars, nanospheres, nanoshells? Nanorods have been shown to be the most efficient at converting non-thermal light into heat. And that's exactly what our therapy does. Heat kills cancer. We do it gently from the inside out. Sona's gold nanorods are unique. They are patented, and they are the only ones in the world that don't use a particular toxin in order to give them the elongated shape that permits us to have an aspect ratio, so that they act like little tuning forks. They've also been rigorously tested by the FDA and National Cancer Institute's nanotechnology characterization laboratory. We've gone through 8 different batches of our materials, have passed through with flying colors. And the NCL, in fact, their personnel have sat with us on our side of the table in meetings with the FDA, which is extremely helpful, and we owe them a great debt of gratitude. So what are we doing with this technology of ours? We've developed a two-step therapy. We call it targeted hyperthermia therapy. The first step of which is to intratumorally inject these inert, gold nanorods into tumors. This is a red blood cell, one of the smallest molecules in the body, just being shown for a scale reference. Those little flecks inside it are the gold nanorods. So once injected in the tumor, we shine our near-infrared light on the tumor. This is a case of melanoma, late-stage irresectable melanoma. Near-infrared light tuned specifically to our nanorods so that they resonate and create heat, passes harmlessly through about 1 to 2 centimeters of tissue before hitting the gold nanorods. The gold nanorods convert that light energy into heat by vibrating, causing friction and causing heat within the tumor, heating the tumor from the inside out. Of course, heat kills cancer. We do it at a gentle temperature of about 45 degrees Celsius, which ensures that only the cancer cells are being destroyed here, and keeping it below 50 degrees, after which, of course, healthy cells start to get harmed. So this is a very selective precision medicine, if you will. And our targeted hyperthermia has two key benefits that we've shown now in our third set of preclinical studies. Number one, targeted hyperthermia can eliminate tumors. We gently heat them in from the inside out, and they shrink down. And here's a great example of one such tumor for which that's happened. But frankly, it was only when we made the acquaintance of our friend, Dr. Giacomo Antonio, who's had a career in immunology, in which we started to have our eyes opened as to the benefits of that slow denaturing of the cell that's achieved when you use hyperthermia being, again, about 45 degrees Celsius. In that case, we're stimulating the immune system and priming the immune system to fight the cancer on its own and to attack metastatic sites. Our goal for THT, as we call it, is to clear the cancer and to prevent recurrence. So I'll now hand it over to Carmen to take us through briefly our past results and then dig into the colorectal data. Carmen.
Great. Thank you, David. So great. So exactly the goals of this therapy, as David said, are to A, clear the tumor through immunity, and B, create a long-lasting effective immune protection. So we've published, this has been submitted for publication and is in review right now. This is our first group. This is a triple negative. That means estrogen receptor, progesterone receptor, HER2 negative breast cancer. And this breast cancer has been typically a very difficult cancer to treat in the clinics. And here we showed early on, and some of you will have seen this data, that when we treated the tumor, these are tumors grown in mice, and these are grown in two sides of the mice, two flanks, if you will. When we treated the tumor with interleukin-2, which is a standard immunotherapy that we use in our clinics for melanoma, and we have a clinical trial in breast cancer, that we had little or no response. So the blue control line is simply saline or salt water. The red line is the interleukin-2 alone. Again, an immune therapy that if there's an immune relationship between the cancer and the body, this will drive it by activating or driving the T cells and the immune cells that are in a tumor to kill the tumor. We added, this is our targeted hyperthermia therapy alone, and we do get a response. So you see that line is starting to come down, and this is after one treatment of gold nanorods injected directly into the tumor to limit toxicity to the animal, and the same would apply to a person, and then activated with the near-infrared light. The mild hyperthermia causes tumors to shrink or not progress in growth. We have to wait until they grow big enough to inject them. But after one treatment and after time, the tumor starts to heal and regrow. The final line is the combination of the two. That's the targeted hyperthermia plus the combination of interleukin-2, which you can see on the graph. We gave three serial treatments to the tumor. And what that did was, once the THT activated the immune system to drive immune cells in, we could continue to push the immune cells along to completion of response. So the combination of without the THT, without the gold, there was no immune engagement. Once the targeted hyperthermia and once the mild heat was present and the tumor started to die, those tumor antigens were exposed, and then the immune therapy that wasn't effective became effective. Next slide. So you can see this is our melanoma model, and this B16 melanoma model is a very challenging model to generate immunity. This is one of those cold tumors, cold meaning that without some external force, immune cells don't actually infiltrate it. And so we see similarly in this, and this is part of our current publication, the control, which is salt water saline, had no effect, and interleukin-2 alone has absolutely no effect on these tumors. When we add the gold to the melanoma cells, and different cells are, so that line is a little flatter than the other line, the 4T1 breast cancer model. Different cells are more or less stressed by moderate heat, fever heat, some people would call it. And so, but we see a very good response, and you see these error bars on the yellow line means that there is a range of response. Some tumors responding a whole lot more than others, but is definitely a response that's significantly different than the controls alone. And finally, again, when we add the interleukin-2, what we've shown is the immunity that we've generated through mild hyperthermia is then enabled to be more robust by driving those immune cells with interleukin-2 or T cell activator. Next slide. So at one of our former webinars, people asked about, sequential, like, can you make this more effective if you gave more heat and more laser? And so we did an experiment where we did double treatments, and certainly, so we get a response. You can see the control where there's no response in this experiment. These are B16 melanoma, no immunotherapy outside of the gold and the targeted hyperthermia itself. Then we inject with the gold and activate that with laser, as you can see there at the time point, and the hyperthermia causes the tumor to shrink. And then without a second treatment, you will start to get that regrowth. And that regrowth is tumor healing, new cells regrowing. But when we give a second treatment, by the second laser, just exposing to a second laser, no additional gold, the residual gold that's in the tumor is sufficient to generate enough heat to yet cause another stress cycle and cell death. So even in and of itself, sequential therapies with targeted hyperthermia can significantly, if not completely, eliminate a tumor. What we know is the real active action there is that immune response. Sorry, next slide, and that's perfect. So the next slide, when we take this B16 melanoma model and we activate it with the gold, and you can see the line. The gold has a very significant effect with targeted hyperthermia, and then you start to see regrowth. And when we've added the group with IL-2, we've completely flattened the tumor. And in this situation, when we combine the two, we saw a superb response where not only did we see the initial tumor go flat, we saw the tumor on the other flank shrink, reduce in size. And so the untreated tumor response is critically important. What that does is confirm that we are not just seeing a local effect from heat and injecting the tumor, we are seeing a systemic effect. These cells are being released from the immune system to circulate through the body, and any cell that has antigen that we've now exposed through the targeted hyperthermia is vulnerable to that immune response. And so this is this vaccine effect that people look for, the abscopal response, the response where tumors beyond our target actually respond to the therapy. Next slide, please. So to prove that this is in fact an immune response generated through the hyperthermia, we took both our 14-1 breast cancer model, our B16 melanoma model, and we took the tumor cells after treatment, and we looked at before treatment, what immune cells were there, what immune signals were being sent, and after treatment, how did that change? And we see that with the gold nanorod targeted hyperthermia, that this is a standard panel on the left-hand side, that column of markers. This is a standard panel of immune markers that would be consistent with active immune activation of the innate system, that system that has to respond first to generate the adaptive response that we're looking for. And so it works. Both of these tumors are, this is critical to the story that the targeted hyperthermia is an immune activator. So these two results were very exciting. These are both cold cancers, cancers that don't typically respond. And we showed that the standard immune therapy, interleukin-2 alone, had no effect. Treatment with interleukin-2 after treatment with targeted hyperthermia was effective because we initiated immunity. So the next cancer is colorectal cancer. And as many of you will know, colorectal cancer is the third most common cancer among us, equal death in males and females. And the vast majority of these cancers are cold. And the definition of a cold tumor is kind of what you're seeing on the screen there. These tumors either have no engagement of the immune system, so there are no insufficient immune cells within the tumor to actually affect an immune response, or the cells that have invaded the tumor have been overly suppressed to the point where none of the systemic checkpoint inhibitors, those PD-1 inhibitors or CTLA-4 inhibitors that we give in other cancers, have an effect. So a cold tumor is not a candidate for immune therapy. The question is, can we convert that cold tumor to a hot tumor, i.e. a tumor that now has immune cells within it that can be further activated, stimulated, or enabled through some systemic treatment like a Keytruda? So the conversion from cold to hot was essentially what we did with the targeted hyperthermia through the Sona Gold nanorod. Next slide. So this colorectal cancer model is essentially the industry standard representative of most colon cancers. And in this model, we do the same as we do with all mice. So it's the tumor cells are injected into the flank of the mouse, and then we see if we can stimulate the mouse immune system. If the tumor is not immunogenic, it'll continue to grow without any effect in a very predictable manner. What you see here is the CT26 colon cancer model, the mouse model. This is the tumor, and we've treated this tumor. It's growing nicely without any interruption from PD-1 inhibition. So the checkpoint inhibitor, the Keytruda or the pembrolizumab, these drugs have no effect, largely because of that previous slide you saw. There just isn't an immune complement in that tumor to make them active. Next slide. Then we add our hyperthermia. Again, the objective of the hyperthermia is to generate mild heat. The mild heat creates a stress death in the cancer cells. That stress death draws innate immune cells into it. And we see that the tumor is arrested. Most of the reduction you're seeing right here is because the targeted hyperthermia has caused the stress death, and the tumor has simply shrunk through an apoptotic cell death. And then you're starting to see some regrowth, as we expect, because there is a healing response to moderate heat. This isn't ablative heat. We haven't destroyed the entire tissue. We've created a stress death in the cells. Next slide. So our question and our hope is that if we add immunotherapy to this, do we achieve what we did in the other two models? And this was really quite exciting to me and to the team. You can see here that when we added the targeted hyperthermia plus the PD-1 inhibitor, now we've chosen a PD-1 inhibitor and not IL-2 because IL-2 wouldn't be applicable to a colon cancer population. We only use IL-2 predominantly in melanoma and other skin cancers, and now in some triple-negative breast cancer studies. But this would be a PD-1 model. So we wanted to make the model reflective of current standard of care. And in this, not only did we see the flattening of the curve that we hoped, but we saw 50% of those animals that responded completely. In other words, there is no tumor residual here. This is a complete clearance of tumor. Next slide. So the question is, THT, what did the targeted hyperthermia do? So and this is a question that comes up alone. Can you just do the gold? Is that enough? And in some cases it is. You see these error bars? That tumor initially responds and they're all tight. So up to day 5 after the targeted hyperthermia, That's a pretty tight situation where most of those cells have gone into a cell death and most of the tumor is essentially annihilated. But over time, there is a range of tumors that will stay small, and that's where the error bars, those whisker bars are on the lower end. Those tumors are not regrowing. And then there's a range where some tumors are regrowing more quickly. And of course, this is the heterogeneity, the spectrum of response we see in biology. And so, but we know within that mix, after day 5, those tumors that are growing also now have admixed in them an immune cell population that recognizes them. And so to enable that immunity that wasn't there before is really the goal. And so you see with the addition of an immune enabler like a PD-1 inhibitor that the tumors are flattened. So the next slide would show that. And so there's the difference. This is gold on its own with a range of effect that is good, but you can't pick the winners. I mean, biologically, again, the heterogeneity of the tumor populations that we deal with are definitely vast. And so what we're capitalizing on is the fact that we have created an immune cell population that's now receptive, and in this model we've proven it handily. So what we know is that colorectal cancers that are typically not responsive to immunotherapy, will respond when activated or enabled through targeted hyperthermia. Next. This is an effective treatment. We're showing it over and over again. This results in a very significant tumor response. A lot of the tumors have shrunken to the point where you don't know if they're going to regrow. And in fact, in some human cancers, we call it watchful waiting, where we will wait and see if the tumor regrows before we go to the next phase in treatment. In this situation, we know we're generating the immunity and we're capitalizing on the knowledge that we have effectively generated. So, but the gold alone, the targeted hyperthermia, results in significant response that is quite shocking. And when combined with THT, we know we've got a winning combination here. This is the 1-2-3 punch. The gold plus the near infrared light is the 1-2 punch, and then you follow through with the uppercut, if that's the right boxing analogy. And this takes the tumor down. It's wildly effective now in 3 clinically cold challenging models.
Thank you, Carmen. And I'll remind folks that we're happy to take questions. There are a few questions queuing up in the chat box on your screen. You just click that chat box and pump in a question if you have any. You're happy to take them now or any other time. But now looking forward to what is next for Sona. This is, as Carmen says, we now have 3 solid preclinical efficacy studies in 3 different types of cancer with 2 different types of immunotherapies and compelling results from all 3. So what is the question, is the point? Well, this, we believe, portends success in the future in going into humans, but that's got to be proven out. So that's, well, our first thing, before I jump ahead of myself here, the next thing coming up, the first 2 studies that were done have been crafted into a manuscript and a paper that has been submitted for peer review, and that publication decision is pending. We expect that soon. That's, we think, going to be initiate a bit of a coming out party for the company, where suddenly we're on the radar in the industry, because right now it's a small preclinical life sciences company saying, look, we've got some great mice results. You've got to have it peer reviewed and appear in a leading journal before it's taken seriously. But at that point, we think that we'll be gaining recognition in the industry and beyond. So we are excited about that. And Carmen and the entire team has done a tremendous job with a journal article, a manuscript that I think everyone will be very impressed by. And the initial feedback from the submission has been very strong. Lots of questions coming in now that we'll get to in a moment. With the publication of that journal article, we'll be initiating we'll have a platform effectively to be able to initiate an investor education program. We're going to be doing more webinars, we're going to do more regular communication, and we're going to do a great deal of outreach coincident with that too. We felt like we needed to have that peer review of our data in order to serve as a platform, a soapbox, if you will, for us to get on and start shouting about this, not just to the investment community, but importantly to the industry and potential big pharma partners in the future, for whom this would be a prerequisite to have any sort of a conversation. The human studies. Obviously, this is what matters. This is our goal. We want to get in. We believe this is going to translate. Happy to take questions on that. And therefore, we are pushing hard to get into, in fact, 2 different studies, and it's a dual track. One we're looking at overseas, one we're looking at in North America, different timing associated with that. But we are pushing hard and making good progress on that now. And we'll look forward to having updates on that as we go forward. Finally, once we have those human data, we don't want to get distracted because we are uber focused on that. But nonetheless, the team, in some of the brainstorming that's done from time to time, we recognize that this is a platform technology, and there are many other applications to it, whether they could be antibody-drug conjugates, diagnostics, etc. Once we have our initial human data, that will give us a ticket of entry, if you will. It'll afford us the opportunity to go off and explore in some other areas and develop a bit of a pipeline of further projects and products potentially in the future. But the focus right now is getting into human and getting those results and sharing those with everyone. On that timeline, development milestones, we've got a goal to be first to have our first in human data through an early feasibility study in 2025, and the goal is to deliver those results within the first half of this year. We're pushing on that. It's tricky stuff. It's not uncomplicated. I kind of skipped over, I think, on the last page, but study design is everything. A well thought out, a well developed, a well laid out study can make all the difference in the world. So yes, we're taking a little bit of time to do that, but we think it can have powerful results, and you don't want to be rushing into these things without having thought through everything that needs to be done. When some of the inclusion and exclusion criteria that are necessary and optimal for the study may be in conflict with each other, and you want to optimize for that. That's the work that's been going on. Our biocompatibility feasibility study is underway right now. Dosing's been done on that, and we expect that, and that can answer some of the questions that have been asked today too. So we believe Sona is in a really strong place right now. We have compelling efficacy data across multiple cancers. We're moving towards first in human data in 2025 quickly. We have a program to raise the profile, and that's going to start with a validated, peer-reviewed perspective on our study results. And then off into the future, we can see leveraging this platform to do other things of interest, other areas in which the core gold nanorod platform can be developed and monetized. With that, I want to thank everyone for the attendance, but first we're going to go to questions, and I've got a number here that I'll pose off. Like this one in particular, Carmen, can you have a situation where you overdose gold nanorods?
That's a great question. Currently, because we're injecting intratumorally, the answer is not systemically, so no, but you can put too many nanorods into a tumor such that you can cause ulceration in the overlying skin in a melanoma situation. So we have actually done, I didn't show any of it today, but we've done a lot of dosing studies, and we've actually reduced our initial dose that we were treating in our first study by 75%, getting the same results and far less ulceration in the overlying skin. Because it is, even though it's mild hyperthermia, if the gold distributes into the skin, it can cause the blood vessels to essentially clot off and cause an ulcer. So it's not a burn per se, but it's an ulcer like a diabetic ulcer would be in a person with bad diabetes and peripheral vascular disease. So yeah, the short answer is no, and the long answer is it's complicated, but you can.
And the safety studies that are being undertaken right now by GLP labs in the US are showing the fact that we are, you know, required to stress test dosage and show what the highest possible dosage, what sort of harm might come from it. And the results look extremely good. It's promising. We're not concerned around that, particularly as Carmen says, because we're injecting them directly into the tumor and not doing systemic administration. Another question here, you've tested on some aggressive tumors. If you were successful bringing this to market, what other tumors can be considered, i.e. glioblastoma?
So that's a great question. You know, the CNS and the central tumors are probably going to be later. The answer is yes, you probably could. If some of you know anything about GBM, that glioblastoma, that, you know, we thought for a long time we wouldn't see an immune response in a central, like a CNS tumor. We do see immune responses. There'd be a lot of work to do in the brain in terms of the safety studies and the collateral damage, because everything is so tightly related. The next tumors after we do breast, in the human, we will be doing melanoma and breast, because triple negative breast cancer is actually underway with IL-2 here in Halifax, and then rectal cancer, because it's readily accessible. And currently, the standard of care for the locally advanced rectal cancer is radiation, which causes enormous toxicity to the patient and eventual dysfunction. And so we're optimistic that this is the better, more effective option in that regard. And then tumors like gastroesophageal junction tumors that are easily accessible through endoscopy. Pancreatic tumors would be very accessible through gastroscopy. So those are the things. And as David mentioned, and I won't go into it because it'll get a little tangential, but the theragnostics. So tumors that are metastatic, that we can combine our THT with an imaging study and drive therapy at the time of diagnosis. This is an exploding horizon for many people, and we're perfectly suited to be a part of that path as well.
So, Carmen, we're getting a question here. Will the colorectal cancer study also be published?
Oh, yes, absolutely. We're excited about that. There's a lot more data I didn't show you. What I wanted to show you today was essentially the overall effectiveness of it. But this is even more elegant science. Lessons learned from our earlier studies. We've done sequential hyperthermia with the immunotherapy in the colon cancer model. And we actually have, because we've seen complete clearance of tumor, we're looking at that to see if that animal is essentially vaccinated. So we've got re-challenging studies underway. So the next study will be far more elegant in terms of both the science and the long-term outcomes. And finally, we have some insights as to those graphs and tumor growth, because it's a healing response. It's a very exciting, that's a cool bit of immunology that people didn't know about, that we're the first to show that healing response is actually part of the natural response that you can, once you understand the biology of it, you can take advantage of as well.
So another question here specific on the graphs. At 15 days, why doesn't the graph show any range of tumor size? It only shows tumors completely eliminated. And that goes to those 4 mice for which their tumors were completely eliminated.
Well, there's just, at that point, there is no, I mean, essentially they're flat. It's flat skin. There's nothing to measure. So there's no range. Some of the earlier data, and that's another good question around the science of it. Some of in the early days, when we were putting the larger dose of gold into the tumors and we were having some skin ulceration, the tumor numbers would be diminished, because the veterinarians we would have to sacrifice the animals for obviously humane reasons. And so that was the right thing to do. But we've been completely forthcoming with all of the data, because these generate good questions that hopefully help you believe that we are, dealing with the challenges and solving the problems as we go along.
To that end, great question here on, can you speak to the team's experience with first-in-human clinical trials and subsequent development through exit or launch? If the team doesn't have direct experience, are they working with advisors, et cetera?
Yeah, so that's a great question. So in terms of intralesional injection, that's been my career for the last more than 15 years, directly injecting tumors. So I will be able to do the teaching to the the PIs about how to inject tumors and what to look for. And through direct injection with immune therapies, a number of them, including interleukin-2, but also using BCG and all kinds of other immune activators, we have a catalog of skin responses and immune responses that we can use in the teaching. The activation of the gold to generate the THT is a skill set that the lab techs have now, and that would be part of the learning curve. That's why this first trial is so important, and that first trial is very simply designed to show that we can inject the gold, activate the hyperthermia, and do it in a well-tolerated way and generate some heat and response. The objective of the first trial is that very thing. So these device trials, as they are, and in fact, the ethics boards expect that this is part of learning curve, and so the first bit of information for first in humans is going to be that learning curve, for sure.
And to the team's experience specifically with clinical trials, you've cited the 2 past trials recently that you've been involved in on breast cancer in particular.
Yeah, so we have 3 registered trials. So in terms of trials development ourselves, we have locally created 3 trials, 2 injection trials in melanoma, and now 1 in triple negative breast. It's an IL-2 trial. It's in clinicaltrials.gov. You can look it up. Greg Knapp is the PI for that one, and we have injected our 3rd patient, and that was a huge. And building infrastructure around that trial was huge. So we're creating a culture of tumoral injection in Nova Scotia that creates this kind of system that we can really take advantage of, because people are of like mind around this now. That IL-2 trial with triple negative breast cancer is the only one in North America, the only one in the world as far as we know.
And of course, for an external study, we'd be engaging a credible, experienced, and medical device specific clinical research organization to aid with that.
Right.
Question here about what sort of anticipated adverse events there might be in human studies that you could anticipate.
So for our trials, the ones I just talked about, the ones are going to be understanding the tumor response in humans. In other words, when I inject tumors with my immune therapy, they ulcerate. We're going to have to understand, is that ulceration part of the treatment or part of the immune response? And so those are the things I think that this is the learning curve of the first in human trial for sure. And so I think that's where we put our thinking caps on and take notes. But I don't see a lot of. I mean, we expect to see tumors to ulcerate as they shrink, as particularly rapid shrinking, and so that's not going to be that. We're almost hoping that that's part of it, and then analyzing those tumors, because we will be sampling and biopsying as we go to understand whether it's a treatment effect or an immune effect. An immune effect would be completely acceptable. If it's a byproduct of the treatment, then it would teach us how to alter the treatment.
Right, in a question here, do you see the possibility of the THT being fast-tracked in the US by the FDA, considering the results so far? And with the new administration being very vocal on getting the FDA to get new drugs and treatments into market much faster than in the past. Well, I think, Karen, go ahead.
That is a I think I'd be it's way out of my scope of knowledge to comment on that.
Yeah, look, the first thing we need to do is show human results. This is and get this paper published. Being published will put us on the radar, and will facilitate some of those conversations at a higher level with other partners. And whether a fast track follows, we'll see it. The goal is do this early feasibility study in humans to get even a handful of cases that would then again open the door to having those conversations. And that's something that there was a question here about financial resources. We were very fortunate to raise $3.1 million in September. So that we still think is going to see us through on our runway to get to those human results. We're pushing hard to do things in a smart fashion and in a quick fashion. And as I say, lay them out properly from the start. So a little bit a go slow to go fast approach, which has been a lot of the setup work that's been done the last 2 months. Another question, do you expect or even indeed need a biomedical senior partner advisor in your upcoming trials? I think that would speak to the principal investigator and the clinical research organizations, obviously, and there are many questions on this, running a clinical trial is a very specific skill set, and there are specialist CROs that do that, and we'll be leveraging on them heavily. Carmen, any points on that?
Yeah, no, yes, again, we're looking for as much experience. There are some other devices that are used that are they're not therapeutic, but they're diagnostic devices that use near infrared light in the clinical arena, and we have engaged at least one of those companies at a scientific level to look for advisors and collaborators in this regard. We're starting with melanoma and superficial cancers so that we can really simplify the moving parts to the study and gain early experience into those challenges. So yeah, that's of course that's part of our playbook.
A good question here on going back to those charts and talking about the instance where the 4 mice had been completely cleared of tumors. Doesn't no range imply that all 8 mice had tumors eliminated? I think this is worthy of a little conversation and delving into those results and specifically.
Yeah, that's 4 of the 8. If you look at it, so that the rest of those mice at that point would be sacrificed. So yeah, going beyond that point, the rest of the mice are that they're finished the study. These ones that are cleared, there was no need to sack, and so we altered. They're being followed. They're still alive today, and so those mice would factor into another study. What we want to analyze these mice to see if there is good memory, tumor memory. So they'll be rechallenged at some point. But that is a group of 4 at this point. It was an original group of 8, but 50% of them had complete response and keep on going. So there are no error bars around
that. Great question here on, what has been the response of the PD-1 manufacturers to the possibility of partnership, alliance, synergism? Carmen, do you want to start?
This opens up opportunities for so many companies, so many producers of PD-1, and so many of these trials. So the clinical trials where people aren't responding, like the lung cancer trials or even the renal cell cancer trials where there's persistent cells, if this we can show, and I expect that we will show, that we introduce new immunity through mild hyperthermia. I think there's going to be lots of industry partners looking at this, where the low-hanging fruit here, and it's going to be in the easily accessible tumors to start with.
Right. And I think, again, the publication, some will say, well, big deal, it's a publication, but it's a peer review. And that's what is required to have those conversations, initiate those conversations, and we will be initiating those conversations, and folks will answer the phone once you've got that as appearing in a scientific journal. Today, there are lots of small life sciences companies out there chasing all sorts of things, doing studies with mice. Let's see the peer review data on that, and that will open the door for us.
I would invite everybody on the call to look for this paper in the coming weeks. That we've published it through a clinical lens, so a lot of these questions and interests are addressed through the paper, and our reviewers have been very strict. I hope you'll be as impressed and excited as I am with that.
Yeah. So a confirmation following up on one of those comments saying, so only 4 mice continued after 10 days. Why is that?
Well, because the rest of them were part of the protocol was to sack them and analyze them. This is not a longevity study, but we actually had to alter it. Once the tumor's cleared, we had to actually get permission to continue on with the mice, because the protocol is written as a termination study, so that we can look at that time point and see what the immunity is, look for the cell populations that have infiltrated, what memory's been generated. But when we had clearance, we were not expecting this to be complete elimination. And so when we saw it, we quickly, and rightfully so, said, let's hold on to these mice and see where that takes us.
And just to provide a little color, that's permission from whom?
Well, you have to go through the animal ethics people to do that.
Right. Okay.
And the vet.
And finally here, how soon would you expect to submit the colorectal cancer study, which is still ongoing at this stage anyway?
Well, I sometime February, I think in honesty, when we get the rest, we've got lots of mice growing and we're looking at it from just different aspects. February's hopeful. You never know what's going to happen with this stuff. We had to actually sack a whole batch of mice because the cells were infected with mycoplasma. It turns out it's a laboratory glitch thing. So I don't want to overpromise on this, but I think by end of February we'll have a manuscript ready for review.
And at that stage, there'd be greater longevity data, presumably, too.
Our next two studies are exactly this. Now we're looking for the tumors that are cleared and longevity, and seeing what percentage of those mice have not only cleared the tumors, but you can't regrow them.
So this takes us back to the concept we conveyed about a year ago about the idea of our desire to create a mountain of data. All these studies will support our submissions to regulators. This is what they want to see. The more, the better. And these are thoughtful studies that Carmen's driving, with each one being additive in their structure and in some of the insights being delivered and the points being supported. So it's great data to have. We're going to continue to drive that forward along with the safety data, parallel tracking with the human trial coming up. We'll be providing more information on that as it comes. There are questions here about, when will you announce who the CRO is on that? We will announce when that's been locked down and when we initiate and when first human is dosed. And of course, as soon as we've got data coming out of it. So thank you, everyone, today. Unless there are any other questions, which we'd be happy to take any other time. We look forward to reporting back, and we look forward to the publication of the journal article once reviewed. And we look forward to keeping you up to date on a very regular basis going forward. Thank you.
Thank you, everyone. Have a great day.